So, yeah, like to introduce our speaker for tonight. It's an honor to introduce you to Professor Kevin Balkwill, who is our SAAB Silver Medal Award winner for 2024. He's currently a emeritus professor at the Wits University, and during his, or at least at the start of his career, he studied at Wits and the then University of Natal, and completed a PhD in taxonomy. And during his career at Wits, he served 10 years as the head of department and 7 years as the assistant dean, and simultaneously served 31 years as the curator of the herbarium there. He has a high publication record in different botanical fields, including taxonomy, conservation, the social uses of plants, and other ecological fields. And he's also successfully supervised 10 MSc students and 17 PhD students. To SAAB itself, he's also made great contributions by serving as a council member, and he's also served as the vice president and president of the SAAB Council. And he was also very instrumental in the South African Journal of Botany in the changeover of editorship when he served as interim editor-in-chief. And besides all these academic contributions, he also grows botany among the general public with his numerous workshops and courses that he provides. And we're very excited to hear more about his work tonight. So let's try to get the screen sharing and see if that will work for the talk. If you don't manage, then me or Elizabeth can help from our side. But Prof. Balkwill, please see if you can share your screen.
Right.
And I just want to mention, yes, and I just want to mention that Prof. Balkwill will keep his video off during the talk just to save bandwidth.
Great, we shared the screen. I'm just trying to, right, the slideshow has begun. Great. So thank you very much. Are you ready for me to start?
Yes, we can see it.
Okay, so I'm chatting about xerophyta in South Africa, and I'm trying to show you why I think the taxonomy is quite troublesome and it's an easy job. Working on it, but I hope you'll enjoy hearing what we have done. Did the slide change yet? Just trying to get an idea of the speed.
Yes. Yes.
Okay, great. Okay. I think one of my first contacts with xerophyta was in 1983 when I collected this specimen on a and you'll see the top label in the middle, which is on the specimen at the University of Natal. I made a note at the bottom that it didn't run in Q Bulletin, which was the revision of the time, and I named it as xerophyta species. And I see that the duplicate that went to Edinburgh, someone named it xerophyta rosea, as you'll see on the bottom label. And then someone, I think it's probably Olive Hilliard's hand, had put some reasons why it didn't really match xerophyta rosea, but that's something I've only seen more recently. Nonetheless, this shows one of the reasons that my interest was piqued in this genus a long time ago. Now, when I was studying at Maritzburg, Olive Hilliard was the curator of the herbarium, and she used to go across to Edinburgh for about 3 months a year. And she used to then spend her time there following up on types, consulting literature, writing papers, and working on some of her long-term projects. And so in 1985, when she was on one of those trips, I became a self-appointed stand-in herbarium curator. And so I was dealing with queries that were raised by visitors who came in. And someone brought in a specimen of the xerophyta viscosa, not this particular specimen, but of the species. And when I was looking through the collection to identify it, I came across this specimen on the right-hand side, which you can see has long stems, and the scales on the stem are rather different from those in Xerophyta viscosa, and the note said that the flowers were white. So I took this out and put it on one side, and when Olive returned from Edinburgh, I gave her a report back on what I'd done while she was away and showed her the specimen. And she took one look at it and said, "I'll have that." And the next year, 1986, it was published as a new species in the Edinburgh Journal of Botany. So that again was me touching on Xerophyta. In 2013, a revision was published. And so I thought, okay, well, now Xerophyta's sewn up, so we can put it to rest. But you'll notice that in the introduction, in the second paragraph, the author said that that as many members of Velociaceae grow in localities that are difficult to reach and therefore botanically unexploited, it is expected that yet more species will be described in the future. What I also discovered when I came to look in more detail at this revision, that other than types, they hadn't borrowed material from any Southern African herbaria to undertake this revision. And so obviously there are going to be big gaps in it. Also then when I was out in the field, I noticed that on the Witwatersrand rond, one would have specimens like you see on the left, where the stems would be single and unbranched. You would have around the Eswatini border these very coarse specimens with very, very thick stems. And then around Nelspruit, one can find these plants that have branching stems, but they're nowhere near as thick as those around Eswatini. And there seem to be various differences in the flowers as well. So I thought this is a potentially very interesting retirement project. And so I thought I would start working on it this year. Now, that was quite a long time ago that I made that decision to do that. Sorry, now that I have, okay. So in 2017, we went on a trip to Legalamitse Nature Reserve and came across this rather interesting Xerophyta, one I'd never seen before. I'd never seen one that made that kind of base before. And so I was totally intrigued. And I was with Barbara Turpin at the time, and I said, this is something I want to get to work on in the future. And she said, well, Douglas McMurtry and Shane Burns are actually working on a revision of the group. So once again, I thought, okay, this is then something I'm not going to work on. But once I had moved to Nelspruit and I had been on some outings with Doug and Shane, and we chatted about various things, in 2021, they actually invited me to join them on this group. So in fact, I started working on it from before my retirement period. So some background on the group is called Bobbejaan Stert, and you will see similar this stem with the particular fibers that are on this stem, how very close to the tail of a baboon. So from that point of view, it's aptly named. And the other common name is the black stick lily. And here again, you will see how we've got these black stems and they produce lily-like flowers. And so once again, this is a very apt common name. In 2011, a paper was published which established that worldwide in the Velociaceae, one should recognize 5 genera. And these genera then occur, one with a single species in China, one occurring in Africa and Madagascar, which is Xerophyta, and then there were 3 in South America. At that time, they estimated about 50 species in Africa and Madagascar, and over 200 species in South America. These plants are resurrection plants, and Jill Farrant is an expert on that, and she has described their strategy as poikilochlorophyllis, and when they dry down, they lose all their chlorophyll, they dismantle the thylakoid membranes, and they go purple with anthocyanins, and then those anthocyanins mask them and protect them from the sun during the rehydration process, when the thylakoid membranes are reassembled and the chlorophyll reconstituted. They occur in habitats which are usually rocky outcrops or where there's very shallow soil, and I think this means that there are low intensity fires and there is low competition, and I think that that is important in their biology. And because of these habitats, the kinds of habitats that they occur in, they are patchy in the landscape. So these populations are growing on various islands of suitable habitats, and there must be relatively little gene flow between the different subpopulations. And indeed, this has been studied in South America. They've looked at the genetic diversity between populations and within populations, and concluded that in this particular species they studied, the different populations may indeed represent incipient or cryptic species. And that certainly is along the lines that we are thinking in dealing with them in South Africa. So the history of the group is that in Flora capensis, there were 7 species, and they were all treated in the genus Valosia. In 1921, Greaves did a revision and recognized 8 species for South Africa and in the genus Valosia. 1974, Smith and Ayensu did an account of the African ones, and for South Africa, they recognized 7 species, 1 of them in the genus Talbotia and 6 as Xerophyta. And that Xerophyta elegans was originally described in Talbotia, so they were then resurrecting that genus. And then in 2013, the Benker revision that I showed you earlier has 15 species and 1 variety, and they are all treated in the genus Xerophyta. Now, traditionally, 3 groups are recognized in the genus in South Africa, the one being Xerophyta elegans, otherwise known as Talbotia, which I'll come back to. Then there is the shrubby group, which is centered on Xerophyta retinervis. It is the herbaceous group centered on Xerophyta schlechteri. So here's Xerophyta elegans, which was previously Talbotia. It has a slightly different vegetative morphology, and it grows in a very different habitat. You'll see on the left here, it's growing as an epiphyte. I don't think, found the others as epiphytes. It grows associated with streams, in the shade, and on mossy rocks. So it grows in a much damper environment than the other species. Now, in this paper that looked at the phylogeny of the group in 2011, what they found was that Talbotia was indeed basal to the rest of the Xerophytas, but they said that for reasons of maximizing phylogenetic information and ease of identification, Talbotia should be transferred and treated as part of Xerophyta, and that currently is how it is treated. However, there are quite a few differences that have been recognized between them. The one is that the ovary is glabrous in Talbotia, whereas most species have either glands or excrescences on the ovary in Xerophyta. We have recently described one species in Xerophyta which has a glabrous ovary. In Talbotia, the ovary is angular and the angles are winged, whereas in Xerophyta, the ovary corners are rounded. In Talbotia, you will see that the style sticks out beyond the anthers, so the style is longer than the stamens, whereas in Xerophyta, the style is shorter than the stamens, or certainly ends before the stamens end. The stigma is globose in Talbotia, whereas it is cylindrical in Xerophyta. The habitat is moist and shady for Talbotia and dry and exposed usually for Xerophyta. In Talbotia, the anthers are extras, shedding the pollen towards the edge of the flowers, whereas in Xerophyta, the pollen is shed towards the center of the flower or from the edge of the anthers towards each other. So those are some quite important differences that were recognized. And since that time, our beginning to work on the group, we've also found that it seems that there are specialized scale leaves that protect the stem in the one that was previously Talbotia, where in Xerophyta, the filaments are attached to the tepals and then become free. In what was Talbotia, the filaments are attached to the roof of the ovary and then go up vertically. And then in what was Talbotia, there's no projection of the connective beyond the tip of the chambers of the ovary, whereas there is a projection of the connective beyond the tips of the chambers of the ovary in Xerophyta. So there are indeed quite a few differences that we've noted that weren't known before. And then there's this paper that was published in 2014 looking at the seed coat development. And they have concluded that merging Talbotia with Xerophyta is also not supported by their results, as these species have quite distinct coat composition. So there is quite a bit of evidence building up to say that we should be recognizing species as, or this species as a separate genus. So one of the things that would make me more convinced that this was something that was wise and helpful would be if there was more than one species in the genus, although I don't think that is necessarily a requirement. So what is interesting about the distribution is that there is definitely a disjunct distribution, with one group being distributed on the escarpment of Limpopo and Mpumalanga, and then there is another distribution in the KwaZulu-Natal, Drakensberg, and then going down quite close to the coast, and certainly nothing is known in between. There are some differences in that most of the northern populations have a very small stigmatic surface, whereas there is a much larger stigmatic surface in many of the KZN populations. But we haven't had an opportunity to investigate all of this in great detail yet, and to decide whether or not there may be more than one species. At the moment, whatever we look at seems to vary between populations within both of those geographical areas. And then another issue which perhaps mitigates against recognizing Talbotia as a separate genus is that someone has actually hybridized a member of Xerophyta with a member of Talbotia. so that concludes then the Xerophyta elegans group, and we go on to the retinervus group. And these are the ones which are shrubs, and they have these very unusual stems, which have stem material in the middle, and then they have roots, adventitious roots growing down around those, and they are bound together with resin, and then they initially have leaf bases around those adventitious roots, and eventually near the bottom those leaf bases may be shed. They also have excrescences or hairs on the ovary, and they are visited by flower beetles. And occasionally we have seen little bees visiting them as well. The Xerophyta schlechteri group is herbaceous, and it has glands on the ovary and on the pedicel. We've seen this being visited by small bees. Very seldom have we seen this visited and eaten by the flower beetles to the same extent. And here's one which is pendulous in its growth habit, but nonetheless, these are ones that we would characterize as being herbaceous rather than shrubby as in the other group. However, there are some that are intermediate. Here you can see what the tip of one of the shrubby one's stems look like. And then we have some within the schlechteri group where there are hardened leaf bases, and you can see that these make little areas that are very similar. The stem tips are very similar to those of the retinervus group. So what are the methods that we use? We engage in lots of field work, which we enjoy, but it can be challenging. And it's absolutely vital working on this group to do field work. You can't do it working on herbarium sheets only, and I'll show you that a bit more as we proceed. Also, Doug and Shane have been collecting plants over a long period, and we continue to do that and gradually take over more and more and more of their shade house, as well as them planting many in the garden, so that we have them growing under like conditions, and that helps us then to be comparing how they would perform under like conditions, and whether or not the differences we are seeing are simply due to the environmental conditions of the particular habitat where a specimen is growing. The advantage then of having these cultivated specimens is that when we are writing the descriptions, we can base them on fresh material. And when we've had a bit of rain, the herbaceous group flower quite reliably, and we have fresh flowers that we can use working on those descriptions. Then we do a lot of herbarium work. This is in the herbarium of Doug and Shane at their property known as Whitethorn. And in addition to herbarium specimens, what we have done is to photograph specimens at the National Herbarium at Pretoria, and at the University and various other places. And we photographed a full sheet as well as details on the sheet. And then we've made one color print of a full sheet of each of those specimens, and we treat that as a herbarium specimen. But when we want detail, we can go to the more detailed photographs of each of those specimens. So we can work on specimens in a number of herbaria without having to have those specimens on loan. Then as the plants flower, Doug and Shane are meticulous in keeping the numbers very carefully of exactly which plant belongs to which specimen, and they will pick off flowers, they will section them, do measurements, and then they will add them onto the sheets. So this provides a permanent record of what they look like when they are in flower. We use JSTOR Global Plants a lot to examine the types as well as types that we have seen and photographed in various herbaria. And then anatomy is very important in this group. It gives very important characters to distinguish between species and groups of species. And so what I did was, for a few years, I offered projects at WITS for third-year students. Mr. Innocent Kambuli was the on-the-ground supervisor of all of the lab work, whereas I was helping the students with understanding what the project was about and helping them with their analyses and comparisons and so on. And so that provided us with an opportunity to have a lot of the species that we had in cultivation sectioned and to get information on the anatomy. Then what I did was to build a reference collection in PowerPoint where I took scans of the various micrographs of the anatomy from many different sources, and then I arranged them into one consolidated PowerPoint so that one could then have two copies open and compare the anatomy of one species with the anatomy of another species much more easily than having to get different references or get hold of slides. So this is a summary of some of how we've set up that reference collection, which makes it possible to compare between the different species, to compare the anatomy. So what are the characters that we look at? One of them is the general morphology. So there's the well-known shrubby form, for instance. Then there are herbaceous ones. There are some that form these domes that we nickname tortoises. And those are in the glandular group. And here they don't form upright stems. This is one which is in the shrubby group, but in fact it only has short stems and has many stems. And then here is one of the herbaceous ones, which is forming long stems which are pendulous down a cliff face. Another character is size. So we have this is Xerophyta burroseorum, which is not only the one that has the glabrous ovary, but it is also the smallest of the xerophytas. There are those that grow to a meter or so tall, well known, but one of those has grown to much, much taller, and we're very keen to try and relocate this plant and see whether it is still alive. It really is something that is quite spectacular. So the full size to which a particular group may attain is something that is helpful as a character. And just on that, I thought it would be interesting to chat about the age that some of these might reach. And so work again has been done in South America, where they've looked in detail at a particular population. They've done dissections. They've found the particular scar stem and worked out how far they are apart. And because that particular species flowers on an annual basis, they can work out how produced in a year, and they can work out the increment in the between when flowers were produced in 1 year and flowers produced in the next year. And so from that, they have looked at about 8 to 12 leaves were produced each year. That translated in a 5.5 millimeter increase in stem length per year. And that means that a plant 1 meter high is 182 years old. And a plant that they mainly worked on was this Velosia colbecki, and you'll see that that is very similar to our Xerophyta retinervis. And so I think it's not too far a stretch of the imagination to think that our plants could be aged in a similar way. So this Xerophyta, currently treated as part of the retinervis group, must be about 5 meters tall, which would mean that it's 910 years old. And these, which are in Eswatini, north of Mbabane, these plants must be about 3 to 4 meters tall at least. And that would mean that they're somewhere between 546 and 728 years old. So this for me is quite staggering, that a plant which has this kind of morphology and this kind of anatomy can grow to an age of this nature. Another character that we would use is whether plants branch or not. So the Xerophyta retinervis around Pretoria and Johannesburg doesn't branch, whereas those around Nelspruit branch freely. The thickness of the stem is important, so that there is a form of retinervis around Barberton, which we fondly call megapoda, which develops a very, very thick stem. There you can see it, and there you can see it, as opposed to others which at similar heights have much, much thinner stems. Here you can see some which have even thinner stems than that particular one. The shape of the tip of the stem is quite important. So the typical Xerophyta retinervis, and this one is Xerophyta vallis pongolana. They will have cuneate or acute tips to the stem, whereas this is one that grows in Queen's River near Barberton, and this is one that's very prevalent in the Waterberg, where the stem broadens out at the tip, and so has a very different shape. The stem surface is important. You can see that here there are short fibers on the stem. Here there are longer fibers on the stem. Here much, much longer fibers on the stem of some in the retinervis group. Whether or not the leaves are persistent or not is important. So here's Xerophyta remanii. You'll see that the leaves are persistent, and once they've died, they remain attached to the plant. Whereas in this particular species, or this one, this is a new species to be described. This is in the retinervis complex. You will see that once the leaves reach their full age, they abscise and are lost. In most species, the leaves are slightly curved or are straight, whereas there are a few species where the leaves are curled, significantly curled. And I don't know whether Stefan is in the audience this evening, but we really need Stefan Sibbert to help us to get to the locality of this specimen that he collected, because it seems it might be different from Xerophyta romani on the basis of having these very curly leaves. Leaf width is a character. So you'll see here's one with a particularly broad leaf. Here's one with an intermediate leaf width, and here's some with narrow leaves. The leaf margin, here's one where there are no teeth on the margin, are pretty sharp, short teeth. And then here one where there are long, wispy hairs along the margin. So other features that we can use to distinguish between them. Some of them have glabrous leaves, top and bottom. Some of them will have hairy margins. Some of them are hairy, glabrous above. Some can be very hairy on the upper surface. And then there's one group that has specific short curved hairs just in one particular place of the leaf. And so these are all features that we can use to distinguish between them. Some of them have no glands in the leaf, and others have obvious glands in the leaf. and that leads to there being exudate. It usually starts out as white, but might end up being quite dark. And those that have the glands that are exuding substances have an interesting herby smell to the leaves. The color of the tepals can be different. So some are always white, some start white and then will fade to mauve, and others open purple and remain purple. So the color of the tepals can be helpful. Then the length of the style, the length of the stigma, and the length of the anthers and the relative length of the stigma to the style are all features that are helpful to distinguish between species. There's one species where the anthers are white instead of yellow, as in all the others. So that's an interesting character for that one. Then the ovary might be glabrous, as in this Xerophyta barrosiorum. It might have glands on it, it might have wispy soft hairs, or it might have these excrescences on the ovary, as in Xerophyta retinervis. And then those excrescences can differ between species. You'll notice that these are unbranched. In this one, they branched. The density of the excrescences might differ in that it is very sparse in the species that occur in KwaZulu-Natal. And then on some of the serpentine patches, there are species where there is a subsidiary excrescence on each excrescence. So again, another feature we can use. The hairs can also be different in that they can be simple or single in some. You can have one or two fused together in others, and then you can have a number of hairs fused almost into scales in other species. And then the glands can vary in size. Sometimes those on the pedicel and the ovary match one another, whereas there are one or two species where those on the pedicel and the ovary are completely different in size and nature. So the glands are helpful features. Then the anatomy is helpful. Basically, there are three types represented in South Africa, but then there are all kinds of variations in the anatomy between the different species, the shapes of these bars, the position and the number of vessels, and many other features help to distinguish between the species. Then there is some correlation with geology, but I think it's largely physical to some extent, they need these special habitats in which to grow. So we found them growing granite, quartzite, norite, ultramafic, and they particularly love dolomite. Now at Legalamice, we found 3 species growing together on one dolomite patch. Then we went on to a shale patch, and there are no species growing at all. Ground, you'll see there's a dolomite patch, and there you can see that there's at least one species growing on the dolomite. So nothing on the shale, but many on the dolomite. On the other hand, when you go to Bivan Dam, there's one species growing on the quartzite outcrop, and then there is a different species which grows on the shale at Bivan, and it only grows on the shale. So in some taxa, they seem to move between different geological formations, whereas other taxa seem to be limited to particular geological formations. Now, doing this work, we have lots of challenges, and one of them is that the herbarium specimens, as you have in the middle here, do not necessarily give you a clear picture of just how different a plant might be in the field. And so that is why it's absolutely essential that we do a lot of field work in this particular group. the plants look very different. So this is the same species on the same outcrop on the same day. The area where this one grows was burnt and this one was not. And so you can see how different the plants may look, whether or not they were burnt in the previous winter. Another challenge is, how do you match the types with the actual taxa in the field? So the types will be represented with something like that, whereas in the field you have a form that is very different from anything else, and it takes a lot of knowledge and actually having seen the plant in the field to be able to realize that, okay, this is probably this particular plant and not any other. So that is one of the challenges we face. And then this is the same species, but now it hasn't been burnt. And you'll see how much more difficult it would be to match this particular specimen to the type specimen because of how different it looks when it hasn't been burnt. Another challenge is that the type locality data can be very vague. So this is the type of Xerophyta clavata from KwaZulu-Natal, and the only clue of where it occurs is Natal. And we know of species that are within this complex from Ganjla, north of Kranskorp, from Kuzi, Jacini, and near Lake St. Lucia. And it's really tricky now for us to decide just which of these different populations actually corresponds to the type. And as you can see, there is not a lot of material to go on the type specimen in any case. Another challenge is that the shrubby group seems to be very sporadic in flowering, and we have not yet been able to predict just when they will flower. Sometimes when a population is burnt, the next year they will all be flowering. Sometimes a population is burnt and then the next season there are no flowers in that population. Sometimes in a widespread population, some patches will be burnt, some patches will not be burnt, and they will both be flowering. Someone has said, well, exactly 3 weeks after the first heavy rain, they will come into flower, but there's some that really don't seem to flower in some years. So this is a challenge of doing the field work and getting the material we need, is that we can't predict when this shrubby group will actually be. The other problem we have is that there is a lot of variability in the field, and you can see here the differences in color and the shape of the tepals. And I think Carl might be in the audience, so thank you to Carl for these pictures from the population near your home. They are very helpful and informative. Another challenge is that we have currents of species. So this is the same outcrop in the Dolomites on the edge of the Volkberg. And here you'll see Xerophyta villosa. Here you'll see another species, which is one of those in the glandular group, which has these tortoise-like bases. And then here is another species, and that is one with long prostrate stems, and that would fall into the Romanii group. So you can have at least 3 and here quite different species growing at the same locality. And then the challenge is, whoops, what happens if there are similar but different species, so genetically isolated species growing in the same locality? Here again, you can see 2 very different species growing very close to one another. And here's a species with glabrous leaves that forms small trees, and next to it is one with very hairy leaves, and this is in Queens River near Barberton. So this is one of the challenges we deal with. And if you can get these very different species growing together on the top of Abel Erasmus Pass. In one patch, you can see how much smaller the leaves are here, how much smaller the flowers are than this one that's growing just here. And so we've collected bits of both of those, put them into the same pot and grown them, and you can see that the size difference is maintained when they are being treated in exactly the same way. So it's then a challenge to decide how do you deal with this taxonomically? Is it or is it not the same species? Is it just variation within the species? This is another locality where I went with Simon Atwood, and here you can see small leaf plants. There you can see slightly longer leaf plants. These small leaf plants had very small flowers. The longer leaf plants had larger flowers, and then there were other plants with even longer leaves, and they had flowers with much longer and narrower tepals that arched back. And again, we've taken some of each, cultivated them in the same pot, and you will see that those differences have been maintained where they are cultivated under identical conditions. There are also some mistakes in the 2013 revision, and that provides challenges that we need to grapple with, and makes it not quite so easy to follow up on the work, so that you will see that a species with bases like this, which would have been the tortoise species, for instance, a species with these long stems and another species with short stems, have been cited all as Xerophyta rosea. And we're not convinced that those are all the same species. There are at least 2, if not 3 species represented there. There are also some mistakes in the labeling of some of the anatomy, in that it is said that this is Xerophyta retinervis, having been sectioned in the dry state, whereas this is Xerophyta retinervis sectioned in the wet state. But the locality from which this one came, it's more likely that there's only Xerophyta villosa at that locality. And if you look carefully here, you'll see that there's vasculature in this area, which is not present here. So it's very unlikely that that vasculature would develop on the wetting of the plant. So I think that this micrograph has been incorrectly labeled. There's another challenge that specimens are misquoted. So one specimen, Sciopus 1034, is quoted here under Xerophyta purpurascens, and here it's quoted again under Xerophyta villosa. So which species did these authors actually treat this specimen as? Then in terms of doing the anatomy, because of these sclerenchyma bars and then soft tissue in between it, it is not easy to get really good, clean sections that one can use to do your analysis. And then there is possible hybridization. So at the site where Xerophyta barroseorum was first collected, described it, we'll have Xerophyta barroseorum with glabrous ovaries. We will have this one in the tortoise group, the ovaries, and then there is a third entity which has glandular ovaries where the glands do not match the tortoise group, and of course it has glands unlike the Xerophyta barroseorum. Now initially we have said this is possibly a hybrid, but we want to go back and do some more field work and ascertain whether this is not perhaps yet another species occurring at that site. Then we have a difference of developmental changes. So this particular species, which is, or entity that is recognized as a variety of Xerophyta reti, when it is young or it has been impacted by chewing by game or by cattle and goats, it will have many stems and the leaves are shorter and broader than at other times. But as it matures, so it will get more longer stems, and there will be quite a few stems from the base. As it matures further, it gets fewer stems from the base, and then eventually a single stem from the base, and eventually it will have this long, naked or unbranched trunk, and then a number of stems near the top. So this is a challenge that you need to be able to realize that in anything we're dealing with, we have to have in mind it might look like one thing when it is young and look completely different when it is 900 years old. And then linking into that, it seems that there might be cohort recruitment. So these are two subpopulations of what we are convinced of the same species. And you will see that in this particular group, they haven't reached the stage where they have branched yet, whereas in this group, where they're of similar age, they are similarly branched because this group is older. And so because you then have a whole group that looks similar and another whole group that looks similar, it's tempting to think, well, maybe these are different taxa. And then the excrescences and the glands and the hairs also change in terms of their structure and in terms of their density as the ovary matures. And so another challenge that we have. Another challenge is that these plants grow very slowly. So the ones on the left are 9 months old from seed, the ones on the right 18 months old from seed. So you can see that thinking that you can collect seed in the wild, plant them and grow them up and get to know something about the plant is a bit of a pipe dream. And then initially both Doug and Shane and then I in my learning thought that we could go and collect small plants and they would grow in cultivation and they would flower in cultivation and give us what we needed. But so this plant was collected at this size and it does flower. This plant was collected at this size probably 10 years ago. It looked exactly the same as the day it was collected, and it is not flowering. And there were plants, it is a different taxon, it is at a different locality, but where this plant occurred, there were other plants that looked like that. So is this going to grow in a few decades or maybe a few centuries into something like this, or will it remain with this morphology? And that because of the slow growth, that is a challenge we have to deal with. Another one is how the material is pressed. So in this case, the leaves were pressed separate from the stem and were in contact with the blotting material, and so they remained pressed with a bit of the stem attached. There wasn't pressure on the leaves, and so they fold up sort of concertina-like, and they appear much narrower on the herbarium specimen. They also look very different if they collected in different as this one would have been pressed while the plant was dehydrated. And you can see then how different leaves appear and thus the specimens appear on how they were when they were actually collected and the specimen was made. Another challenge is the flower beetle. So sometimes we're very lucky to find 2 or 3 flowers or 2 or 3 plants that are flowering in a population, but then the flowers are mated by these flower beetles. And even when there are many plants that are in flower in a population, it's quite a challenge to find some flowers that haven't been damaged by the flower beetles. I'm not sure whether or not these Flower beetles may be the pollinators. We have occasional little bees visiting, but this would make a nation project for someone, except for the not something be suitable for a because area. Another challenge is where the plants grow can make them rather difficult to get to. You can see Shane here on this cliff face, Xerophyta barrosiorum, and it was quite a challenge. Shane is the one who usually ends up and then the type locality of A. xerophyta is the summit of Mamatswiri, and we set off to try and collect it. Unfortunately, we planned to leave later in the morning than we should have, and one of the people walked along the top of the ridge. That's where we got by the time we absolutely had to turn back, and that is the summit of the mountain where the type specimen was collected. And so we do not know what that specimen looks like, and we will have to go back, which will not be altogether unpleasant. So where are we now? We have a working list of probable South African taxa with reasonable consensus. So we have 3 people who are quite strong-willed, who are prepared to speak their mind, and sometimes we have 3 very different opinions on what each plant is, but we are getting to the stage now when we get new specimens or new pictures of specimens that we look at, we can pretty much say, well, that belongs in such and such a species, and we agree in a relatively short time. So this is our list of putative species in the herbaceous group, and you'll see those that are in bold are the ones that are currently described. So there are quite a few on which we need to do quite a bit more work. And this is our list of putative species of the shrubby group. And again, those that are in bold are the ones that have been struck through, are ones where we have decided that they actually belong in something else. Or we've changed our mind and decided it's not different from the others. So with this probable list, we think there are possibly 20 to 25 new herbaceous species for the country and possibly 18 to 23 new shrubby species to describe. There is 1 species to be resurrected, and there's 1 variety to elevate to species level. We have now formally described 1 species, Xerophyta barrosiorum. So it's always useful to have that under your belt. And we now have developed a strategy, which is that we're going to deal with the herbaceous species first, get that sewn up and published while we're looking at the others sort of in the background, and then we'll move on to the shrubby ones, which are a lot more challenging. We have compiled a template of to help us write the description, very many other descriptions, and looked at all the characters that have been described for species of Xerophyta and compiled that into a template that we use when we do the descriptions. We have written descriptions of some species, and we have started to list the specimens for some of the taxa. So we are making progress towards getting ourselves finished. So thank you to Neil Crouch, who made many, many contributions to this work. He started out with Doug and Shane when they started on the group. They were often helping him find other material, and together they discovered a lot of very interesting xerophytes. So thank you to Neil Crouch for all of his contribution. Thank you to Delia, Simon, and Jan, and many others for assistance in the field, but especially Delia, Simon, and Jan. Thank you to Ini Kambuli for all his very hard work on the anatomy projects that were done by the third-year students at WITS, and to the many third-year students at WITS who took this on as a third-year lab project. Thank you for your contribution. Then thank you to the Herbaria who've loaned us material and facilitated access and made it possible for us to do photographing. And thank you to the conservation authorities and landowners for permission to do the field work and to collect the material we need to do the work. And thank you very much to all of you for listening.
Thank you very much. That was fantastic. Thank you very much. That was great. I think you certainly chose a good group to work on that have taken you to beautiful places. We will take questions now. And I'll start off with the question in the chat, but please feel free to raise a hand if you have any questions. So John Moore is asking whether of all the xerophyta species and the potential new species, how many have been confirmed as poikilochlorophyllus and are desiccation tolerant, basically? Do you know how many?
Yeah, okay, you can hear me. Yes, they all are. It's something that is characteristic across the family. So even the Xerophyta elegans, or the one we prefer to refer to as Talbotia, which grows in the much moister conditions, that also desiccates, and it's one that goes very, very purple with the anthocyanins, and then it resurrects. So it's something that is characteristic through certainly all the South African ones.
All right, thank you. I was wondering about the effect of fire on these plants, since it seemed like some of them occur in grassland ecosystems, right? Do the stems of these plants survive the fire, or does it get partly killed? And does the fire stimulate multi-stem growth in them?
Okay, so I mean, initially I thought that they would be fire resistant, something like you see in Buafani, where the bulb has all of those very fine the compressed leaves, the old compressed leaves around it, and it's referred to sometimes as the telephone book effect, if you young people know what telephone books are. Right, even although it's all that very thin paper, because it's so close to each other and there's no air that can get into it. So I thought that it was that, and they were fire resistant. Now, what's interesting when I was doing the reading about the age is they mentioned that in South America, some of the stems are chopped up and use fire lighters because of the resins that are in the stems. So they have resins, and those resins are to some extent flammable. So I think part of whether is on these rocky outcrops, or where the soil is shallow, because then there's not going to be a high fuel load and the fires are going to be less intense. However, fire does seem to stimulate some populations to flower. The Xerophyta retinervis does survive fires where they are not too intense, but especially what's quite interesting is the terminal bud and the terminal rosette seems to be very important in holding that stem together. And where plants have had that terminal rosette eaten, the stems start to collapse. or where it's killed by continuous browsing, maybe I should say. Those stems will collapse, and then that is very, very vulnerable to fire. But I have, I mean, the areas in the Waterberg where the survival rate is less than 1%. Now, whether there was something else that caused it and then a fire, or whether this was an absolutely massive fire that went through and caused that level of kill, I don't know. But certainly I have seen them damaged by fire, and I've seen some that I think are probably have been killed by fire. Although others are fire resistant if it's not too intense.
Very interesting. And as William Bond also says in the chat, they're strange plants. So Eugene Moll is also asking, what do the genetics show about this group?
Okay, so okay, I'm not quite sure what he's asking there. We haven't done the genetics. I did approach the South American group to see whether we could send them specimens or material to do DNA on our South African group. And they then asked me, well, how many species? And at that stage, I had no idea of what answer to give them. So we haven't proceeded with doing more detailed DNA of the South African group. There is that South American paper that I cited, where they have found that within populations, the variability is relatively low, but between populations, the divergence is quite high. And we have been trying, I've sort of been holding little carrots at Kelsey that I would like her perhaps to come and look at these populations where we have these ones which seem to differ by size, to see whether that might be to do with polyploidy. So that would be another aspect of the genetics that would be quite interesting. But we certainly haven't looked at genetics, and I would hate to try and tackle this group deciding on what species they are and aren't by doing DNA of every single variant from every single locality.
Right. Thank you for that. Please let me know if there are other questions. I was wondering about the plasticity. I think that the nursery experiments you were doing are very interesting. Which characters do you think showed the most plasticity? Was it just in leaves and flowers, or what do you think they're most flexible in?
Yeah, so again, this is something that perhaps we haven't, we're only tumbling to. So when we were looking at the group that it's proposed to call Xerophyta macmurtrii, and we started looking at specimens and at the cultivated plants, we noticed that there seemed to be variation in the width of the leaves. And then we said, okay, well, maybe this is to do with the areas that they are being cultivated. So there was one that was being cultivated on the rockery, and another one from the same locality, original locality, was being cultivated in a pot. And then when we went and got leaves from the two, they were identical. So I'm not sure. And yeah, it's something that is very difficult, I think, to try and study, is how much variation is there within the same genotype, and how much variation is due to different genotypes. So there is a lot of genetic variability between them, I think. And then, but they don't seem to change that much between the condition in the field and then under cultivation. And when they are different in the field and we cultivate them in the same pots, they might look sort of fresher and so on, but they maintain. If the one had longer leaves in the field, it will have longer leaves in the pot and so on. So they seem to maintain those differences in cultivation.
Okay, thank you very much. There's one question from Tsepiso online who wants to ask whether your research group has ever specifically focused on medicinal plants, or your research area has only been focused on investigating xerophytes, and if so, what's the significance? So I'm not sure if it's asking whether you've looked at medicinal plants in xerophyta or in general.
Okay, so in the past, I have supervised students who were looking at the sustainability of harvesting medicinal plants. I've never looked at medicinal properties of any particular plants. Now that I have retired, I want to just do my first love, which is taxonomy, going out into the field, finding species, deciding what they are, and where we're lucky enough to find new ones to describe them. And so xerophyta, that is what we're doing. That's the taxonomy. In terms of the relevance, if someone was going to do studies, for instance, I mean, the interest, really, really fascinating work that Jill Farrant has been doing is to see whether you can transfer these resurrection qualities to crop plants. So that drought would not affect crop plants to the same extent. Now, if you've got 13 species or you've got 60 species, the number of species you should be looking at for variance in the genes to perform that is very, very different. So from that point of view, I think there's helpfulness in looking at the taxonomy. But then also from a conservation perspective, if one thinks that Xerophyta retinovus is one very widespread and variable taxon, as is what is currently thought, as opposed to, we think it is probably 20 different taxa that are very restricted in their distribution, and some of them will be are likely to be threatened. I think then from a conservation perspective, there's a lot of relevance to what we're doing.
Great, thank you very much. And then Patricia Holmes asks why you're only looking at the plants in South Africa and not Southern Africa, because surely the tropical species will help to solve some of the taxonomical issues.
Okay, so again, what I have now started doing is looking more carefully at the stuff from further north in Pretoria and looking at those specimens and seeing how they compare. So for instance, it was thought that Xerophyta villosa occurs in Zimbabwe and South Africa. The type is actually South African, but when I looked at Pretoria at the Zimbabwean material of villosa, the hairs on the ovary are completely different. So it's not the same taxon occurring in. Also with what is regarded as being Xerophyta porky, the name at the moment, but there's one which is recognized as being a species that occurs in Zimbabwe and so on. Now that we've looked in more detail, ours is different and needs to be described as a different species. So we tended to regard that maybe the 2013 revision was a good account, and all we needed to do was the South African ones. The more we look, the more we see that maybe there's more to be found in further north. But it's absolutely essential to do field work on the group. You absolutely can't revise a group like this working only on herbarium specimens. And that is just beyond what we'd have time to do and what we'd have finances to do. So I think to sort out the South African ones would be a big contribution. But yes, indeed, we will examine all the known ones from further north before describing new ones from South Africa, which might actually already have a name further north.
All right, fantastic. Thank you. Jan Graf was just saying that you're probably thinking of Xerophyta ramosa.
That's correct.
Right.
So that's what it's regarded as. It occurs in the north of Limpopo and down into the Kruger Park. And it was regarded as being the same as Porky ramosa, which is from further north, but it's different.
All right. Well, that was certainly very interesting, and I think it's time to wrap up. Thank you very much for an excellent talk. This is really insightful, and I think the botanical community as a whole is delighted that you're continuing with this taxonomic work. So I do hope that you enjoy it and that it's very fruitful, and that you still have many years of these adventures.
Well, thank you very much. Thank you for the opportunity. Thank you to those who have listened, and thank you for the interesting and helpful questions. I appreciate that, all of that. So thank you and thank you to you and Elizabeth for your organization, and especially for our trial yesterday, without which this would not have worked. So thank you very much to you.
Fantastic. Thank you very much. Andrew, please have a look at the comments in the chat. There are several compliments for you. So I hope you have a good evening further. And yes, thank you again to the audience for listening, and I hope you enjoyed it. We will have some more of these webinars next year, and this webinar is or was recorded, so it will be available on the website of SAAB, so you can access that there in a couple of days. But yeah, hope all goes well with everyone, and we look forward to seeing you at the next SAAB webinar or at the actual SAAB conference. Good evening.